Hi, everybody, and welcome to Mr. Mitchke's online Biology 30 lessons. I hope my students are staying healthy and safe and doing well. This is an unprecedented situation that we have never been through before, so we are going to do our best to help you guys through your courses, and I'm going to be providing tutorials and lessons online, and I'm just going to get started right here with lesson 1, which is our last topic in our genetics unit. So last time we left off, we had done chromosome mapping. If you missed that lesson, make sure you review the chromosome mapping section on my Moodle notes under linked genes and do the practice problems in the booklet. And the answers to those answer keys will be posted on Moodle for everything, so just keep that in mind. What I'm going to do now is attempt to show you the booklet while I'm doing this video. So I'm still figuring out some of the ins and outs of this video conferencing technology called Zoom, and I'm going to just do my best here. All right, so hopefully we can see the document here. This is the booklet. We're on the section called Genetics and Society. In the space below, list different examples of how humans have used selective breeding to acquire desirable traits in both plants and animals. So you can list any examples you can think of where humans would be selecting desirable traits in either plants or animals. Just some ones that might come to mind right away are agriculture in general, gardening, growing plants, selecting plants that we want to have those desirable traits, bigger, juicier fruits and vegetables, for example. And in terms of animals, we've been doing this through the domestication of all sorts of animals. Dog breeds is a good example, selecting desirable traits like physical characteristics that we want to see in the offspring, as well as just livestock, bigger animals for more meat, leaner pigs and cows, as well as horses for horse racing. So these are just some examples of these instances where humans are selectively choosing traits so that hopefully the offspring will also inherit those desirable characteristics. I'm going to go to my PowerPoint here. Remember, this is all available on Moodle. So we went through some examples of breeding plants. Corn, domesticated corn is another example. Pretty much all domesticated fruits and vegetables are a lot different from what they used to be in their non-domesticated state thousands of years ago. Canola is a Canadian example, which was developed in 1970 as a new type of rapeseed that was not poisonous to animals. So the original plant was called rapeseed, yes, rapeseed, and it developed into canola through selective breeding of all the plants that had the least amounts of toxins. So that created canola. The can part of it is Canadian, so it's kind of a cool Canadian example. These are some animal examples that we went through already. And yeah, dog breeds. Really interesting that all domesticated dogs are the descendants of wolves. So thousands of years ago, humans started interacting with wolves and basically adopting them as pets. And then through selective breeding, all of the different dog breeds we have today are from those original ancestors. The main focus of this section is human genetics and how we trace genetically inherited traits in people, because we don't do experimental breeding on humans for obvious reasons, not ethical at all. So we use different tools to track the inheritance of traits through generations. Also, humans wouldn't be a good genetic study organism because we only produce very few offspring and we live for very long periods of time. So far different from the fruit flies that Morgan used, which were like the ideal genetic specimen to use. What scientists use is what's called a pedigree chart, which is a chart made of symbols that shows the pattern of trait inheritance in families. So this is the main focus of this section, and this is the last thing you need to be proficient with analyzing in the genetics portion of Biology 30. These are the symbols that are used in the pedigree charts. So some key ones to highlight: males are squares, females are circles. If there's a line between two individuals, that means they had a mating. There's mating between close relatives. Yes, something that is frowned upon in breeding, but it did happen a lot in royal families in hundreds of years ago in like Renaissance times. Then we have Roman numerals that denote the generations, and regular numbers indicate individuals. So these would be two parents, male and female. They had 3 children in the next generation. Oops, sorry. Go back there. Identical twins, non-identical twins. This is an important one. If a shape is shaded in, that means that individual has the disorder or has the trait. So be aware of that one. These ones aren't actually used on your diploma, but they are in the textbook, I believe. Known heterozygotes, known carrier of an X-linked recessive trait, deceased individuals, and sex unknown. So come back to this legend to review your symbols at any point. Starting off with what are called autosomal dominant pedigree charts. This is when individuals are affected by the disorder if at least one dominant allele is inherited. So in other words, these individuals will have the disorder or the genetically inherited trait if they are homozygous dominant or heterozygous. Now, when we look at these charts of autosomal dominant traits, dominant disorders appear in every generation in at least one child. So we're going to have at least one affected individual in each generation. Secondly, if one child has a recessive phenotype, this means the parents must have been heterozygous for the dominant trait. In other words, we can have a male and a female who have a disorder, and they can produce unaffected children if they are both heterozygous. So we'll come back to that with some examples later on. These are just some disorders that are inherited through a dominant pattern, autosomal dominance. Polydactyly, which is extra fingers and toes. Huntington's disease, Marfan syndrome. Huntington's is a nervous system disorder, and Marfan syndrome is a connective tissue disorder. Okay, so here's our first look at a pedigree chart. Again, the Roman numerals state which generation it is. The numbers represent individuals. So here are the 2 original parents. Think of them as the grandparents in the family. They had 3 kids. So any vertical lines connected down from them are children of theirs. Now this individual over here, 2-1 and 2-5, those are not children of these people. So just be careful with that. These are people that like married into the family or had kids with their original kids. So we have the grandparents up here, we have the next generation, and then the next generation. Now, autosomal dominant characteristics, we should see a shaded in shape in each generation, and we do. There's shaded in individuals in each generation. And we can also have a situation where 2 affected parents, like the grandparents here, can produce an unaffected child. Now, if this happens, we know the grandparents here are heterozygous because this one child would have to be homozygous recessive. So we'll come back to some of those specific genotypes in the example problems. Let's go on to the next one, which is autosomal recessive pedigree chart. Individuals are affected if they have 2 recessive alleles. In other words, individuals that are homozygous recessive have the trait or have the disorder. Remember, those are the shaded in ones in the chart. So when we have an autosomal recessive trait in the pedigree chart, we have fewer shaded in individuals in general. Usually it's only seen every couple of generations, so the trait or the disorder can skip generations with a recessive trait. Affected children may have unaffected parents. So an affected child might have 2 unaffected parents only if those parents are heterozygous. PKU and cystic fibrosis are 2 disorders that are inherited in this fashion. So here we have a autosomal recessive pedigree chart. Now, the first thing to recognize is there's a lot fewer shaded in shapes than the dominant chart that I just showed you. So we only have 1, 2, 3 shaded in individuals. Those are individuals with that disorder or that trait. We can also see that it has skipped generations. There's no individuals with the trait in generation 2 and 3, and then it reappears in generation 4. So that would mean, again, these parents would have to be heterozygous. So that allele gets carried on from one generation to the next, but the actual trait doesn't show up until here when those individuals would be homozygous recessive. The last one is a sex-linked trait, and remember, most often these are X-linked characteristics because of the size of the X chromosome as compared to the Y chromosome. And we learned that more males are going to express the trait or have that disorder than females. Males only have one X chromosome, so they only need one recessive allele. Females can be carriers but not express the trait. They're able to pass it on to their children. Hemophilia, color blindness, some of the examples we looked at previously in the X-linked section of genetics. And then there's a couple other ones. Duchenne muscular dystrophy is one as well. So here is a pedigree of an X-linked trait that's been passed on through these generations in a family. Now this is from a royal family, which is why we have mating between close relatives. Not recommended. The key characteristic of this chart is that the majority of individuals are males. So all of these individuals that are shaded in are squares, which are the males. The only one is Victoria up here, that's a female. But if you see a large amount of squares shaded in, you can assume it is an X-linked disorder. Now that's the end of my PowerPoint for that section. So what I'm going to do is I'm going to attempt to show you one of the practice problems in the booklet using my phone. So just give me a moment to do that. And I'm going to work through the first example problem in the booklet, and the rest of the booklet is example problems of pedigree charts. So it is just a lot of practice with looking at these pedigree charts, and the goal of each pedigree chart is to determine each individual's genotype. So let's go to the booklet. Just give me a moment to set that up. Here's the bell. Sorry, just one moment, please. Sorry for the delay. I've attempted to figure out how to connect the video to my phone, so I believe I can do that right now, and I will be able to show you the booklets. Of course, it's not going to work. There it goes. Okay, so hopefully you are able to see through my phone camera here, and I'm going to go to the pedigree chart practice problems. Here is the first one. A curved hitchhiker's thumb is recessive to a straight thumb. The following pedigree traces the presence of hitchhiker's thumb in a family. Identify the phenotypes and genotypes of all the people shown in the pedigree. Whose genotypes can you not be certain of? So here we have a pedigree chart practice problem. The goal of any pedigree chart is to identify the genotypes of the individuals. So what I'm going to do first is come up with a lettering system. Capital T is straight thumb. Lowercase t is hitchhiker's thumb. And I know it's recessive from what is mentioned in the question. So I know it's an autosomal recessive disorder. Now, even if I didn't know that, just by looking at the pedigree chart, I would infer that it is autosomal recessive because it has skipped generations. There's only 3 affected individuals, and there's not an abnormal amount of males that would tell me it's X-linked. So definitely autosomal recessive. Now, the blanks below each shape are where you're going to write in the genotypes. So if it's an autosomal recessive trait, the shaded in individuals are going to have homozygous recessiveness as their genotype. So I can start by filling in all of the shaded in individuals with little t, little t. That's the only option for the shaded in individuals. Now, unshaded in individuals could be homozygous dominant or heterozygous. So what I suggest you do is write a big T there, but just leave the other one blank. And we're gonna try and solve any of those genotypes that we're able to. Okay, so you can see I've just filled in one capital T for the unshaded individuals. Now I have to try and figure out some of these missing alleles, and I can do that on a few of these individuals. So if we go to this family, generation 3 and 4, we have 2 individuals that are unshaded, so they are either homozygous dominant or heterozygous. Now they have produced 2 affected children, so that means each of the parents needs to be big T little t or heterozygous, because they donated those little t's to this girl and this boy. I'm going to come up here now. We have a parent who's homozygous recessive. That means all of her children are going to have a little t as well, because she would have given that to her children. So now the other remaining individuals we're actually not sure of. Now this one up at the top, lots of students jump to the conclusion that it should be a big T here because all of their kids are heterozygous. But we have to keep in mind this is probability. So we could have a little t here. Now it's less likely that they would produce all heterozygous offspring if I had a little t there, but it is possible. So we actually have to leave that one blank. We're going to draw a question mark there. Same with this individual over here. Could be homozygous dominant or heterozygous, as well as this individual. And these 2 down at the bottom are all unknowns. And now I'm done. So you have to fill in your pedigree charts like this. You have to be certain of any genotype. If it's not absolutely 100% sure, you can't fill it in. All right, Bio 30s, that's the end of my first lesson video. And the rest of the pages in the booklet are practice questions for pedigree charts. So work through them, check your answers on Moodle. If you have questions, you can email me at my RV Schools Gmail account. And we're gonna get through this together. So more to come. I wish you guys all the best. Bye for now.